All right, ladies and gentlemen, as you can probably tell from this magnificent AI-generated exploding DNA molecule, this is a video about genetics, and in particular, making sure that three individuals involved in this debate are up to speed on the arguments. And those three individuals are Dr. Rob Carter from creation.com, Donnie Badinsky from the Standing for Truth channel, and my dear good friend Jeffrey Tomkins from the Institute for Creation Research, or ICR. And this video is in response to Dr. Carter's video from a few days ago, where the topic of the live stream was chimp and human similarities, or dissimilarities, as I'm sure the creationists are keen to emphasize. Now, I was super keen for this live stream because a few months back I published this video, which pretty comprehensively dismantled every paper that Jeff Tomkins put out over the last decade, trying to push that overall percentage DNA similarity down from the evolutionists' accepted figure in the high 90s down to the mid 80s or even as low as 70%. And in the trailer for Rob Carter's live stream, he said that he was going to contradict not only some of the claims that he had previously made regarding human and chimp DNA, but also some of the claims that some of his colleagues have made, which I took to be a reference to Jeff Tomkins. So here I was, lotion at the ready, keen for some hot yak on yak action, but alas, I was cock-blocked by the fusion of human chromosome 2, which made up the bulk of the presentation. And the flaws in Tomkins' percentage similarity papers weren't really addressed. Anyway, so here is what the evolutionary family tree looks like for the great apes. We have the orangutan on the left, which has 24 pairs of chromosomes, the gorilla also with 24 pairs, the chimp with 24 pairs, but humans with only 23 pairs. Now, obviously, if we just lost a pair of chromosomes along the way, that would be pretty catastrophic as a species. So the inevitable question is, did two of our chromosomes fuse? And if so, do we find evidence of that in our genome? So here again is the evolutionary hypothesis. However many millions of years ago, we have the common ancestor to all four species with 24 pairs of chromosomes. That branch splits off. One of those branches leads to the orangutan. The other branch, which still has 24 pairs, splits again. One of those branches lead to the gorilla, which has 24 pairs. And the other branch leads to humans and chimpanzees, which at that point in time would still have 24 pairs. Then the final speciation happens and you end up with 24 pairs of chromosomes in the chimpanzee, but modern day humans have 23 pairs of chromosomes. So we hypothesized a fusion in the human lineage. So when Rob Carter says the hypothesis is that two ape chromosomes fused, that's not quite as accurate as it could be. It's more accurate to say that two human chromosomes fused. Anyway, let's have a listen to Dr. Rob lay out some claims.
If you look at just human chromosome 2, which is on the top line here, you can see that the chimpanzee, which is the next two down, there's two chromosomes that line up with human chromosome 2 and they overlap at the ends. And there's two gorilla chromosomes and two orangutan chromosomes. The striping patterns are beautiful. Now, we're told that what created human chromosome 2 was an end-to-end fusion of two ape chromosomes. The problem is it's not an end-to-end fusion. The two ape chromosomes are too long. You have to lose a couple million letters there. Something got deleted. So this was a very complex thing. And so if things got deleted, it's a mystery of why the ends of the chromosomes are supposedly in human chromosome 2.
All right, so here's a zoomed in version of what he's talking about. You've got the single human chromosome 2 at the top, and then below that are the two homologous chromosomes in the chimpanzee lined up underneath. And you'll see that they line up really nicely, except for these end bits, which are too long. And here's a similar diagram from one of Jeff Tomkins' papers from a few years back, making quite an effort to align them as poorly as he possibly could. And just read that last sentence. Note the size discrepancy, which is about 10% or 24 million bases based on the known size of human chromosome 2. And he is correct. If you look at the latest assemblies, you'll see that chimp chromosomes 12 and 13, which correspond to human chromosome 2, are about 24.7 million base pairs longer in total. Now, why is that? Well, if any of these three guys had looked into why that is, their research should have led them back to this paper from way back in 1994. So that's more than 30 years that we've known what that difference is. So just to read the pertinent sentences from the extract, one of the significant unresolved differences between the karyotypes of humans and African apes is the presence of positively staining G bands at the ends of many chromosome arms in the chimpanzee and gorilla, but absent from human chromosomes. And then the satellite is more abundant in gorillas and is not detected in humans or orangutans. Now, would you believe that this table took me about a day's worth of programming to build? And what it shows are the various features of chromosomes that we are interested in. So in red, the centromeres. Obviously, if two chromosomes join end to end, then we would expect that joint chromosome to have two centromeres. And we'll show that shortly. But to be honest, I was pretty lazy with the centromeres here. I only searched for the human centromere sequence across all the species, when in reality the sequence differs not just across species, but even across chromosomes. But the results are good enough to show where the centromeres are, so I left it at that. In blue are those satellite DNA sequences referred to in that previous paper from 1994. And as it said, they are absent in the orangutan and absent in the human. And they are way more prevalent in the gorilla than they are in the chimp. There are a lot of theories about why it isn't in the human genome, and some of those theories even involve the fusion. But for the purposes of this video, we just need to accept that it is a brute fact that it does not appear in the human genome. And in green, which you can't really see at this resolution, are the telomeres at the end of each chromosome. But don't worry, we'll zoom in a second. And you'll see I've arranged the human chromosomes to show the two corresponding chimpanzee chromosomes next to each other. So the question is, how well do these two chimp chromosomes line up with the human chromosome if we account for the fact that the human genome does not have any of this blue satellite DNA? The answer is really frickin' well. First of all, you can see that the bottom chromosome has been flipped around, but that yellow line here aligns the active human centromere with the active centromere in the chimp. Well, on the right-hand side, you'll see that really faint red line. That's the cryptic centromere in the human chromosome, and that lines up with the active centromere in the chimp chromosome. Then hopefully you can also see the faint green line in between those two on the human chromosome. That is the fusion site. And you'll see that if you extend that line down through the chimpanzee chromosomes, it lines up really, really well after you account for the fact that the human does not have that particular satellite sequence. Now, you guys are quite welcome to make an argument about how it's maybe not possible, for some unknown reason, for humans and chimps to have structural differences like this. But just be aware that that's not an argument against the fusion. So the fusion involves two human chromosomes, not two ape chromosomes. So the fusion does not require a loss of 24 million bases. All right, let's move into Dr. Rob's next claim.
And if they join together, you should get backwards ones and forwards ones, just like we see there. But it's a very short segment. You're talking like millions of letters got reduced to a few hundred letters, and all the repeats, well, they're not perfect. They're obviously scrambled and changed, and man, would have to have changed very quickly.
All right, and another quick clip.
Okay, and the truth is chromosome 9 has a telomeric-like hexamer sequence, and its reverse complement arranged head-to-head
Yep.
in a similar fashion, but purported fusion site. But isn't this due to a fusion? Thoughts?
That's exactly what I was referring to earlier, and thank you, because I was literally read your paper just recently and watched part of your video on this again, just to refresh my mind on it. Yeah, there's telomere-like head-to-head stuff in the genome that shouldn't be there, and there it is. So it's not abundantly clear that this chromosome 2 is a fusion event.
Well, it is abundantly clear. Let's have a look at that. All right, so these are the basic claims made in those two clips, and I have to say they reveal sort of just how simplistic and completely lacking in nuance their understanding of the fusion is. So the first two claims here are from Rob Carter. The first one basically just expressing incredulity, shall we say, that the fusion signature is only a few hundred letters long, while the corresponding chromosomes in the chimpanzee are 24 million base pairs longer. Well, hopefully now they understand that the fusion did not require the loss of millions of DNA letters, since that satellite sequence just isn't found in humans. And the hypothesis is that two human chromosomes fused, so there's no reason to suggest that that sequence was there in the first place. And just to be clear, I'll state again, there are a handful of different hypotheses about that satellite DNA, which I'm happy to discuss, but not before you guys get caught up to speed on what should be expected at a fusion site. But I'll also steelman Rob's statement a little bit here anyway, just by changing millions of letters down to tens of thousands of letters. And the reason I do that is because human telomeres are, on average, about 10,000 letters long. So if two chromosomes fused, you might expect there to be about 20,000 letters worth of this head-to-head arrangement of TTAGGG up against CCCTAA. And his second statement suggests that these imperfect repeats had to have changed very quickly. So let's address these two things now and then address chromosome 9. All right, and to understand why all three of these objections are silly, you need to understand the three puzzle pieces of how our telomeres work and how those pieces interact with each other. So hopefully by now you already understand what telomeres are, given that you're watching a video about chromosome fusion. But if you don't, then in very simple terms, telomeres are the end caps of chromosomes. When they function correctly, they stop chromosomes from joining together at their ends, which is what we call an end-to-end fusion. And the first puzzle piece, the molecule that gives it that protection, is called the shelterin complex. What it does is bind to that specific telomeric DNA sequence, so TTAGGG, and curls up the telomeres into a ball. So the tips of the chromosomes are sort of tucked safely into bed and not out on the street corner looking to hook up with some other random chromosome. The second piece of the puzzle is called the end replication problem. The way your cells replicate is by a molecule called DNA polymerase, and its job is to make a copy of a strand of DNA. And there's a slight problem though, in that it cannot copy a strand of DNA all the way to the end. There are some cool animations on YouTube as to why exactly that is. Just search for end replication problem. Anyway, the side effect of this incomplete copying is that every time our cells replicate, the telomeres get a little bit shorter. And if they're completely depleted, then you might start to lose genes near the end of your chromosomes. And that would obviously be catastrophic. And that's where the last piece of the puzzle comes in, called telomerase. That's the molecule that sort of wanders around the cell and attaches to chromosome tips that might be swaying in the breeze. And what it does is adds a bunch of telomere repeats onto the end of it. So this is like a very brief explanation of one way that we maintain genetic stability over time. Now, what happens if telomerase fails? Well, obviously there's no method to replenish the telomeres, so they will get shorter each time they replicate because of the end replication problem. And eventually, after a bunch of replications, there won't be enough telomeric DNA for the Shelterin complex to attach to, which leaves the tips exposed and susceptible to fusion. And 3, voila, we have a fusion. And this isn't just a hypothesis. These fusions have been induced in a lab by exactly this method. There's a study from 1997 where scientists disabled one of the components of telomerase in mice, and then over the next few generations, they noticed that the offspring started to have shorter telomeres, and then after a few more generations, the mice started to have end-to-end fusions in their genomes. And this kind of kills 2 birds with 1 stone. So not only does it support a highly plausible mechanism for how this fusion happened in humans, but it also destroys the idea that end-to-end fusions are deadly for the organism. And just quietly, end-to-end fusions being deadly to the organism is something that I addressed way back in February of 2017. So like I said, you boys need to catch up. So what should we expect under the fusion model? Depleted telomeres. So if the telomeres were of a healthy length, then the Shelterin complex would do its job, which is to protect chromosomes from fusion. So anyone expecting thousands of perfect repeats at a fusion site is simply unaware of how telomeres work. So getting back to what Dr. Carter said, we see a few hundred DNA letters of imperfect repeats head-to-head at the fusion site. Well, why is that, you might ask? And to answer that, we need to dig right down to the DNA level of human chromosomes. So here is the actual DNA sequence of one of the human chromosomes, and in this case, it's the end of chromosome 4. And I've just chucked it into Microsoft Word and highlighted all the TTAGGG sequences. But what you can see here is at the bottom half of this page, which is the tip of the chromosome, is a bunch of near-perfect TTAGGG repeats. But as you get away from the tip, so higher up on this slide, you'll see a bunch of imperfect repeats scattered in amongst the sequence. So just to reiterate, this is not chromosome 2. This is chromosome 4. So nothing to do with a fusion. And these imperfect repeats are simply what we find on our chromosomes adjacent to the telomeres. And here is the other end of that same chromosome. So the tip of that chromosome is at the top of the page. And of course, we see the same thing, a whole bunch of perfect repeats followed by a bunch of imperfect repeats. So under the fusion model, where we expect the telomeres to be depleted, what should we expect to see at the fusion site? A bunch of imperfect repeats head-to-head against more imperfect repeats. And that's exactly what we see. So no, Dr. Carter, they would not have had to change very quickly. That DNA was already there.
And if they join together, you should get backwards ones and forwards ones, just like we see there. But it's a very short segment. You're talking like, millions of letters got reduced to a few hundred letters. And all the repeats, well, they're not perfect. They're obviously scrambled and changed, and man, would have to have changed very quickly.
And I'm sure it would be to the surprise of absolutely nobody, except Rob Carter and Jeff Tomkins, obviously, that I addressed this way back in 2016. All right, so the final claim is that Donnie from the Standing for Truth channel found a sequence on chromosome 9 where there are forward and reverse telomeric repeats in a head-to-head arrangement. And his argument, I presume, is something along the lines of, why don't evolutionists claim this as a fusion, as if we're committing some kind of special pleading fallacy for the fusion in chromosome 2. So here is that actual sequence. You've got 4 TTA GGGs on one side and 3 of the reverse sequences on the other side. And if your understanding of the fusion is, simplistic and completely lacking nuance, like, Rob Carter, Donnie Badinsky, and Jeff Tomkins, you might think that this could be mistaken for a fusion. But hopefully now you've seen what we expect from a fusion and why we expect it. You'll understand why this doesn't look like a fusion. We don't expect a bunch of pristine, perfect repeats at the fusion site, because if there were a bunch of pristine repeats at the fusion site, then we would expect the shelter-in complex to do its job and prevent fusion. This idea of expecting thousands of intact repeats at a fusion site is completely 180 degrees opposed to what we actually expect. We expect to see a bunch of imperfect repeats because that's what's right next to the telomeres once they are depleted. But you might be wondering why this chromosome 9 sequence exists at all. I mean, the chances of it existing by pure random chance in the genome are pretty slim, and I agree. And this is a little bit funny because Jeff Tomkins himself cites 3 papers that talk about why this happens. So the first one at the top by Lin in 2008, Endings in the Middle, Current Knowledge of Interstitial Telomeric Sequences. The second one, which is the highlighted one, Contribution of Telomerase RNA Retrotranscription to DNA Double-Strand Break Repair during Mammalian Genome Evolution. And the one below that, Telomeric Repeats Far from the Ends, Mechanisms of Origin and Role in Evolution. And a very brief summary is that occasionally chromosomes break somewhere in the middle for whatever reason. And there happens to be some telomerase floating around that adds on a few TTA GGG repeats onto one of the ends. And you'll see in our genome literally dozens of times where it looks like this has happened. What looks like happened in chromosome 9 is that there happened to be 2 telomerase molecules floating by when this break occurred. So when they rejoined, they were in a head-to-head arrangement. No one in the evolutionary community that understands the evidence believes the sequence in chromosome 9 casts any doubt whatsoever on the fusion in chromosome 2. This fusion just isn't controversial in the scientific community. So I think that'll do for this video. I've really only addressed a handful of the claims, so hopefully we can dispense with these newbie objections and get into the fun stuff like the DDX11L2 gene.
Bye bye, Diaper.
Bye bye.
Bye bye.
Bye.
Bye bye.